So we recently published this using MCL inhibition. So obviously MCL inhibition is important because when we think about BCL2 inhibition with venetoclax-based therapies, one way to develop resistance to it is upregulation of MCL-1. So to inhibiting MCL-1 inhibitor or the MCL inhibitors that would work on that pathway would be important to overcome sort of the resistance of HMA. The problem is MCL-1 is expressed in the heart, so there could be off-target effect. This was always known when MCL-1 inhibition went into clinical trials. And this trial that we published was actually a very fair data reporting on people's cardiotoxicity on this drug, and the drug development was discontinued because of the troponin elevation and cardiotoxicity that was seen on the drug. And it was important to report that because what we actually found, interestingly, is that half of the patients with leukemia, relapsed leukemia, they had troponin elevation even before they went on to the study. So we underestimate how much cardiac stress there might be in patients who have relapsed refractory AML. And when they get MCL-1 inhibitor, we saw in the trial that there was a rise in troponin after the drug, after each dose. And that's why the trial was eventually discontinued. This is a good learning lesson and something that other either direct MCL inhibitors or indirect MCL inhibitors, they need to take into account, build trials that measure these, because it's important to understand this cardiotoxicity. This is the reason why some drugs may not see the light of approval, because either this was not designed well to study, and you have to really know your drug and how it's actually affecting the heart, because that is going to be a problem in the future. And I think it's good to report this because a lot of people are having drugs that maybe the direct MCL inhibitors are somewhat discontinued, but there are other drugs that are in the pipeline that might be inhibiting, having an indirect effect on MCL-1.